Good morning. Good morning, everyone. Happy Wednesday. It is lovely outside right now. If you haven't had a chance to go out, it's in the 60s, the high 60s, very comfortable. Should shape up to be a beautiful day. Yes, if you would, go ahead and type your name or your nickname into the chat for me. That's the way I take attendance. Turn your webcam on and mute your audio. Remember, you're always free to unmute yourself if you would like to speak, or you can communicate over the chat. But I do like to see everybody. I do like to see your smiling faces. All right. We're still waiting for a couple of people, but I'm going to go ahead and get started because we have a busy morning today. So if you didn't see yesterday, your lecture exam is graded and your lab homework is graded. Do take a look at both of those. I was very pleased with the lecture exams. A's and B's across the board, good job. I do make comments on your exams though, so take a look. See if there's any comments that I left. Even if you got an A on it, go ahead and take a look because there might be something that I've written there that you would like to add into your lecture notes. The other thing you might notice about the lab homework is that I'm getting a little bit picky about how I grade. We are at a point now in the course where I'm going to hold you to a higher standard now. We have been learning a lot of vocabulary. We've been learning a lot of, terminology, and we've been learning the difference between certain terms that you might otherwise think you can use interchangeably. So if you type into an answer a term that you're using incorrectly, I'm going to call you on it now. Okay, it's not to be unkind. It's just to help us put into use all of this terminology. The difference, for example, between a sample that we collect from a patient on, say, a cotton swab and a culture that we make in the laboratory, where we put cells into a container with culture media with the intent of growing more of those cells. Okay, so you're going to notice that as we're moving forward, I'm being a little more strict with you about how you word your answers. And I will tell you if you've used terms incorrectly by putting a comment underneath. Okay. Again, it's not intended to make you feel bad or anything. It's intended to help us get our language up where it needs to be as we learn more and more about microbiology. Now, moving into next week, we have finished another portion, another unit of the course. We're moving now into more physiology of microbes. Okay, we're moving into more of the metabolism and the functionality of microbial cells, as opposed to their structural features, their general anatomy. Now we're going to talk about a more deep contextual material regarding how those cells function. And we're going to be spending a lot of time talking in lab, particularly about metabolism, because we have the ability to test in the lab for what kinds of metabolic reactions these microbes are doing. What kinds of metabolic functions do they possess? That's going to help us identify them. Remember, our big job in a diagnostic lab is to take a sample, either from a patient or from the environment. And if we see microbes in that sample, to identify them, to find out what genus and species of microbe that is. And one of the ways that we do that is through what we would call biochemical testing or metabolic testing. We're going to test to see what these cells are capable of in terms of how they survive. What chemical reactions do they use to produce their energy, their ATP? What kinds of sugars are they capable of breaking down? How do they handle proteins? What are they doing every day in order to stay alive? And can they switch? Can they switch from one kind of reaction to another? If they're pushed, if they're put into an environment that is less than friendly, maybe it has lowered oxygen levels or high carbon dioxide levels, how do they adjust to that? Are they able to adjust to that, or will they die? These are the kinds of tests that we're going to be talking about over the next several weeks. And we test this way in order to figure out what these microbes are capable of metabolically. That's going to help us identify them. If you go into a microbiology textbook or you go onto a website that's designed around the discussion of microbiology, there are all kinds of charts and tables that will list the metabolic abilities of all of the microbes that we are aware of. So, for example, we know exactly what E. coli is capable of metabolically. We know what kind of oxygen levels it needs, and we know what it does when it's deprived of oxygen, and we know what kind of media it likes to grow in and on. We know what its gram status is. So when we do these tests in the laboratory, we're gathering data that we can then take into those texts, into those websites, and we can find which genus and species we're handling in the lab by matching up the results of our tests to the known results for that species. It is possible in 2021 to take a microbe, extract its genetic material, sequence that genetic material, in other words, figure out the order of the bases, and then put that data into a database and immediately know what species we're dealing with. It's possible to do that. But it's also very expensive to do that still. It's too expensive to do that. So we still use biochemical testing for the most part, metabolic testing for the most part, to identify microbes in a diagnostic laboratory. There are research laboratories that use genetic sequencing, but for the most part, in diagnostic labs, we don't. It's just too expensive right now. There will come a day when that kind of work, that kind of sequencing work, becomes much more affordable. It's already orders of magnitude more affordable than it was, say, 10 or 20 years ago. But right now, it's simply easier and much less expensive to run metabolic tests instead and gather data about our unknown microbe and figure out what we have. What's the genus and what's the species? All right. I'm just responding to a message here. Give me just a sec. Remember, if you need to message me privately during one of these labs, feel free. You can send me a direct message that will just come to me. Feel free to do that, no problem. All right. So today we're going to start talking about some of this testing that we do. We've already talked about gram status, determining gram status. And yes, that's a critical piece of information to know about a microbe. But what we're going to start talking about today is some other ways that we can test metabolic abilities of microbes. And that's what we're going to be focused on now through the rest of the semester or the rest of the summer. What we're going to talk about today is what we call special media. Now, when you hear that term in microbiology, what we're talking about is culture media. But it's not the kind of culture media that we've been talking about so far. So far, we've talked about culture media as being just a collection of nutrients that we know these microbes need in order to grow, in order to divide and multiply. So we talked about nutrient media, for example. That's a great example of a chemically defined culture media that's designed for growth. Lots and lots of microbes will grow in that media. Special media is different. Special media is designed to give us some information about what these microbes can and can't do metabolically, because special media either adds ingredients or removes ingredients that certain microbes need. So on special media, we are purposefully looking to see what can grow, because if it can grow on special media, it means it has some very particular requirements for growth. So we're going to spend time today talking about a couple of types of special media, why we use them, and what they tell us about a microbe that's capable of growing on them. Remember, we said microbes need certain things. All microbes need water. They need carbohydrate for energy. They need protein as a carbon source. And they need a list of micronutrients. Those things have to be in media or nothing will grow. That's just what a cell needs, basically, to survive. But we can start tinkering with the recipe by adding in other ingredients that might be toxic to some microbes, but other microbes don't mind it. Or we might take something out that a whole lot of microbes need, but other microbes don't. And then we look to see, can this microbe from this patient, can it grow on this special media? Because if it can, we now know something about it metabolically. Did it not grow? That's also important information, because we know what should grow on special media and what shouldn't grow on special media. So we start doing some detective work today. All right. Yeah, I'm getting a couple of messages from people letting me know what what's going on in their lives this morning. And if you need to step away from a lab for some reason in your life, that's fine. Absolutely. I appreciate you letting me know. I want you to let me know. But I also want you to know that that is always fine, because I recognize that sometimes things happen in your life that you can't control. One of the big ones is kids. So believe me, I understand. And just, I will say just generally, as always, if you do have to step away from lab for a few minutes or for the whole lab, just remember that there's going to be a recording of the lab on our playlist, and you're going to want to watch that. Okay. All right. So what you should see on your screen right now is the title slide for our lab today called Special Media. And this is a list of our objectives for the day. The first thing we're going to do is talk about 6 different categories of culture media, just in general. We've already talked about a couple of these, haven't we? We've talked about chemically defined media. We talked about complex media. We talked about transport media. So we're going to add in 3 more categories of media and why we use them. Then we're going to start talking about these special media. We're going to differentiate what's called selective media from what's called differential media. And then we're going to run a little experiment. We're going to take a few microbes, select microbes that we know the genus and species of, and we're going to grow them, or try to grow them, on 2 kinds of special media. 2 very commonly used types of special media. One is called Mannitol salt agar, or MSA, and the other one is called eosin methylene blue, or EMB. But let's start by talking a little bit more about culture media and the different categories of culture media that we have available to us in the microbiology lab. Remember, culture media can be liquid, it can be a broth, it can be solid, which is an auger, or it can be semi-solid. We haven't seen any semi-solid media yet, but we will in the coming weeks. When we make culture media, we are adding ingredients to water. That's going to give us a broth. Or we're adding ingredients to auger. That's going to give us a solid media. The way that you make a semi-solid media is by adding less auger. And again, we'll look at some semi-solid media later in the course. Sometimes when you're creating culture media in the lab and you're looking up recipes for how to grow certain types of bacteria, sometimes you'll find a broth recipe, but you need a solid media. You want to be able to grow colonies. Maybe you want to examine colony morphology, or maybe you're interested in counting colony forming units. So you have this broth recipe, but you need solid media. Well, all you have to do is add auger to the broth, and you will create a solid media. So if you recall from the last time we were talking about culture media, we said there's chemically defined culture media, and then there's chemically undefined, or what we call complex media. Those are the first 2 categories. If a medium is chemically defined, that means that we know exactly what's in it, molecule by molecule. If we are making a complex media, we are adding in an ingredient that is very complex in terms of its components. We're adding in something like blood or egg, maybe egg yolk, some kind of meat extract, something that is a very complex natural ingredient. There are lots and lots and lots of culture media out there. And again, you can buy them pre-made, or you can make them in the laboratory by yourself with a recipe. Most microbiology labs make their own media because it's less expensive. All you need is a recipe and the ingredients on the list. Now, lots of culture media are going to allow virtually any microbe to grow. Remember, we talked about that nutrient media. That's a very commonly used media just for growing microbes. So we're not really thinking about the specific needs of a microbe. We're not trying to differentiate one microbe from another. We just want to grow them. Something like nutrient media would be a good choice. Remember, we do have media that specifically encourage bacteria to grow, and we have other media that specifically support the growth of fungi. Remember Sabarodes media. But a lot of media will allow both of those microbes to grow. Now, there are a long list of microbes that are hard to grow in the laboratory. We give those microbes a name. We call them fastidious. Any microbe that has very particular needs in order to grow them in the lab, we refer to those as fastidious organisms. It just means that they have particular nutritional requirements or they have particular requirements for incubation that make them difficult to grow. In comparison, other microbes are very easy to grow. We refer to those, of course, as being non-fastidious. It's helpful to know which kind of microbe you're trying to grow, but oftentimes we don't know. So when we get a sample in from the environment or from a patient, we often begin with media that will allow non-fastidious organisms to grow. And if we're unsuccessful with that, then we will say, oh, I think I have a fastidious organism here, so I'm going to try some different media. When it comes to the special media, some are designed to support the growth of one kind of microbe, maybe one genus or one species, while inhibiting other types of microbes. Remember, Sabarodes agar is designed to inhibit the growth of bacteria. It allows fungi to grow. It has the nutrients that the fungi need, but it also has a low pH. It has an acidic pH, and bacteria don't like that. So Sabarodes is an example of one of these special kinds of media because we've done something very specific to allow one type of microbe to grow while inhibiting another type of microbe. Now, the other thing we're going to see when we talk about the special media is this point down at the bottom. The way we use certain special media, the way we know whether or not a particular metabolic process is occurring is by using what are called indicator dyes. We love indicator dyes in microbiology. Now, if you don't remember what an indicator dye is from chemistry class, this is the kind of dye that will change color under certain conditions, and it's usually conditions of pH. So the dye will be one color if it's in an acidic environment. It'll be a different color if it's in a neutral environment or a basic environment or alkaline environment. Now, why would it be in a changing pH like that? Well, most of the time it's because this microbe, as it's been growing, is performing some metabolic pathway and secreting some kind of a product molecule. And that product molecule is changing the pH. The indicator dye will tell us, in other words, if a certain product molecule is being produced because it will change color. We'll be able to see it just by looking at the container. And we'll be able to say, oh, look, this microbe is secreting something acidic, or this microbe is secreting something alkaline. The indicator dyes allow us to see that. So let's go through these categories of media. Now, I do want to note that a couple of them go by different names, which can be a little challenging for us, but I'll try to mention all of the different names that they go by. So, a basal media is a media that supports the growth of most non-fastidious bacteria. A basal media is also chemically defined. Okay, so those two terms, basal media and chemically defined media, mean the exact same thing. They have a known list of ingredients. There's nothing in there that we don't know what it is. And it supports non-fastidious growth, so bacteria that are generally easy to grow. Our nutrient media, the nutrient broth or the nutrient agar that we've talked about, that's one of these basal media. There's another kind called peptone water. Remember, peptone is a word we use to describe partially digested protein. So these are two types of media that we use in the lab to grow non-fastidious bacteria. So these are the kinds of media that you would grab off the shelf when you're just trying to see what you can get to grow. Enriched media is the same as complex media or chemically undefined. So these are the media that have things like blood and serum and egg yolk and things like that. These are the media that we add in these very natural but very complex ingredients that sometimes will give bacteria that little extra special nutritional need that they have. So we often reach for the enriched or the complex media when we're trying to grow a fastidious organism. I've listed a couple of the more common enriched media down here. There's one called blood agar, and the name gives away what special complex ingredient it has in it. There's a related one called chocolate agar. No, I'm afraid it does not contain chocolate. Instead, what it contains is cooked blood. So blood that has been heated up so that it starts to change in its character, its chemical character. There are some organisms that will grow better with cooked blood versus raw blood or fresh blood. And of course, when you cook blood, it changes color. It becomes sort of this chocolate brown color. And that's how it got its name. Another very common complex media is called LB. Now, LB stands for something. It stands for Luria Bertani broth, but everybody calls it LB. And LB is just a complex mix of ingredients that can help fastidious bacteria grow. On this slide, on the upper right-hand side, you're looking at an agar plate that contains blood agar. So this is a media that has this one unknown sort of mysterious complex ingredient in it, which is blood. And we know that this plate has fresh blood in the agar because it has that lovely cherry red color. Down at the bottom, this is a chocolate agar plate. So this plate, the agar has blood in it, but it's blood that's been cooked, and it turns that chocolatey brown color. This right here is Luria Bertani broth, or LB. Look at how boring the broth is. It doesn't have any pretty colors associated with it or anything like that. A lot of broth media has this very boring beige color. Now, one of the reasons that I put the Luria broth up here is because I want you to notice the difference between the tube on the right and the tube on the left. When we grow microbes on an agar plate, it's very easy for us to know if we've been successful. We're going to see colonies on the agar plate, or at least streak lines of growth on the agar plate. It's very different when we grow in broth, right? We're not going to see colonies in broth. So how do we know if we've been successful? Well, the broth that we use in the microbiology lab starts off as a very clear, very transparent fluid. In other words, if I took a piece of paper that had writing on it and I held it behind this tube of broth, I would be able to read the words through the broth. It's very clear. Now, after I inoculate this broth with cells and I put it into the incubator for at least 24 hours, it's going to look like this. You see the difference? This broth is cloudy now. I know that I've been successful at growing cells in here because the broth has changed from clear to cloudy. That's my signal that I've been successful at growing cells. So we have basal or chemically defined media. We have complex or enriched media. The third kind of media is called selective media, and this is one of the special media. Selective media is going to allow the growth of desired bacteria while inhibiting the growth of unwanted bacteria. Selective media is almost always agar. That's just something to note. There is something called a selective broth, and it goes by a second name of enrichment media. I really hate that term. And the reason I hate that term is because we just talked about enriched media. So you can imagine it's very easy to get confused between enrichment and enriched. I don't use the term enrichment. I don't even want you to know it. I want you to call all types of this kind of media, whether it's agar or broth, I want you to call it selective media. Selective media is going to allow us to select for certain kinds of bacteria to grow. It's going to allow us to select for one or two or several kinds of bacteria and select against the other kinds of bacteria. Now, one of the selective media that we're actually going to talk about in our exercise today is this one called mannitol salt agar, or MSA. This is one of the most common selective media that are used. Now, MSA has one secret ingredient in it, and that's sodium chloride. This is just plain old table salt, right? This is what we use on the table to salt our foods. Table salt, sodium chloride. Remember, there are lots of other different salts in the world. The term salt means something in chemistry. It means a combination of a positive and a negative ion joined together with an ionic bond. So we don't use the word salt, we use sodium chloride here. That's the particular kind of salt that's in this media. It turns out that bacteria don't like salt. You probably already knew that, right? Human beings have been using salt to preserve foods for centuries, right? Think about the pirates of old that used to bring hard crackers, hard bread on board their ship, and they would salt it to make hardtack. If they fished during the voyage, they would haul the fish onto the boat and they would just bury them in salt. It didn't make for a very pleasant tasting food item, but it sure kept the bacteria off, didn't it? They didn't have refrigerators back then. For most of human history, there were no refrigerators, so they needed means of preserving food out in the open environment at normal environmental temperatures, and salt is a great way to do it. Most bacteria cannot survive if there's a lot of sodium chloride around. So in mannitol salt agar, we can add either 7.5% sodium chloride, or we can add 10% sodium chloride. Most of the time, we're adding 7.5%. Now, what's that going to tell us? This is going to select 4 Staphylococcus species, especially Staphylococcus Aureus and Staphylococcus Epidermidis. Those are 2 species of Staphylococcus that will happily grow in 7.5% sodium chloride. Just about every other bacteria will be killed by that amount of sodium chloride, especially things like Streptococcus. They are very sensitive to salt. I will mention that at this percentage of salt, you can get a very weak growth of 2 other types of cocci. One is called Enterococcus, that's a genus name. And the other one is called Micrococcus. These 2 organisms can survive in 7.5 sodium chloride, but they don't survive well. So if I were to put Enterococcus onto an Msa plate, and I came back 24 hours later, I might see growth, I might not. It depends. It depends on the particular strain that I'm growing. And if it does grow, it's going to be little tiny colonies. It's not going to be the size of colony that we're used to seeing. And that's because even though the Enterococcus can survive in that amount of salt, it's not surviving well. It's under attack by the salt. So yes, it can sometimes grow, but it's going to grow poorly. You're not going to get a nice, beautiful colony. You're going to get a little tiny baby colony that's not very healthy. Enterococcus, Micrococcus are 2 genuses or genera that can survive in 7.5 sodium chloride, but they don't survive very well. Staphylococcus doesn't even notice the salt. It just grows fine. And those 2 species are the ones that we're going to think of: Aureus and Epidermidis. If we see nice, healthy, normal colonies on MSA with 7.5% sodium chloride, we're going to think, oh, it's either Staphylococcus aureus or Staphylococcus epidermidis. Okay, Connie's talking about fermentation. One of my favorite topics. Connie says, I used to make a lot of lactose fermented food. Delicious. Sauerkraut, delicious. And the amount of salt is crucial. That's exactly right, Connie, right? You got to get your salt correct if you're trying to ferment something. And that's because you want to allow some organisms to live, but you don't want to allow other organisms to live, because some organisms will spoil your fermented product. That's exactly right. So salt is one of these molecules that affects the growth of bacteria. And we can play with the level of salt in MSA in order to select for some organisms and against other organisms. So when would I use MSA? When would I reach for MSA? let's say I get a patient sample and the physician tells me that this particular patient had a skin abscess. And the physician is suspicious that this might be a Staphylococcus. That's a good time for me to reach for that media because I know the only thing that's going to grow on it is a Staphylococcus and two very particular species. So you have to use the information you have in order to choose when to use special media and which media to use. I want to know, does this abscess have Staphylococcus in it? Is that what's growing in this abscess on this patient's skin? I can grow on MSA and get a pretty quick answer. So far at least, I don't know which species it is. I don't know if it's Epidermidis or Aureus, but at least I'll know that it's a staph and I can report that back to the physician. Okay. Now remember, we can use 10% sodium chloride in MSA. You can add either percentage. If you go to 10%, the only thing that is going to grow on that is Staphylococcus aureus. It inhibits virtually everything else. So you might say to yourself, well, why not just use 10% sodium chloride? Why bother with the 7.5? Well, what if my physician is asking me, or what if the physician suspects that it's Staph epidermidis? If I'm trying to find out if I have Staph epidermidis, I don't want to start with the 10% because the 10% will kill Staph epidermidis. But good old Staph aureus loves 10%. It will grow just fine. You won't even notice a difference in the colony size. It just ignores it. It's fascinating. Now, a couple words about Staph aureus, because we're all interested in medicine and healthcare. There are different strains of Staph aureus. There are different strains of all bacteria. Now let's review what that word means. Every bacterium has a genus and a species. A strain is the next subcategory. The best way to think about a strain is to think about it the way we talk about dog breeds or cat breeds. So it doesn't matter if you have a German Shepherd and a Chihuahua, those are both the same species. Those are both Canis familiaris. If you don't know what that genus and species is, our beloved dogs belong to Canis, that's the genus, familiaris, that's the species. So they're both the same species, but obviously when you look at your German Shepherd and your Chihuahua, they're very different organisms, and that's because of their breed. So bacteria also have breeds, but we call them strains. Okay, so there's genus, there's species, and then there's strain. There are lots of different strains of Staph aureus. Many of those strains are perfectly harmless. In fact, we all have Staph aureus on us right now. You and I both have Staph aureus on us. It lives on our skin. It also likes to live in our nose, and it likes to live in the back of your throat, back in your pharynx area. Believe it or not, about 25% of us carry the pathogenic Staph aureus on our body, but it doesn't harm us. About a quarter of people walk around every day with a pathogenic strain of Staph aureus on them, but it doesn't bother them because genetically they're just not susceptible to it. There are lots of different strains of bacteria, and within any species of bacteria, there are going to be some strains that don't bother us, and then other strains that might be pathogenic. Pathogenic Staph Aureus is a bad organism. Yeah. You've probably all heard of Mrsa, MRSA. It's a plague in our hospitals and our long-term care facilities. MRSA, or Mrsa, stands for methicillin-resistant Staphylococcus aureus. This is a type of bacterium, a Staph aureus, that is resistant to virtually all of our antibiotics. And patients who have Mrsa are hard to manage in the hospital because you have to be very careful not to carry that organism from that patient to your other patients. So we often isolate those patients in different parts of the ward. We have different PPE requirements when we're handling those patients. It's a bear of an organism. It really is. And there's right now, all we can do for a patient that is suffering with a Mrsa infection is use some of our least commonly used antibiotics. We're hoping that Mrsa doesn't evolve and become resistant to all of our antibiotics, because if that happens, those patients are on their own, basically. Their immune system is the only thing that stands between them and death at that point. There are bacteria in the world today that are resistant to all known antibiotics. And that number of organisms is on the rise. There are, for example, strains of the organism that causes tuberculosis that are resistant to all of the antibiotics. It's very scary. It's a scary thing to think that we could go back to a time before we had antibiotics. But it's in the nature of evolution for organisms to change over time. And these bacterial strains, they evolve in a way that allows them to resist the effects of these antibiotics. Carrie's talking about hot tubs and having a group of people into a hot tub before it was properly sanitized, and having people come down with bad skin infection. Yes, yes. Oh, hot tubs are notorious for that. Listen, you can have a perfectly clean and disinfected hot tub, and you can safely go into a hot tub. But if you think about a hot tub, it's a perfect environment for microbes to grow. It's gross. I know it's gross. So early in the morning. But it's got heat, it's got water, and if humans are sitting in it, it's a great way to spread pathogens. Oh, that must have not been a pleasant 16 cases. Oh, disgusting, disgusting. So yeah, be careful whose hot tub you get in. Make sure that you know that the person who has the hot tub is doing a really good job disinfecting the hot tub. Oh, goodness. Okay. Now, one of the things to know about mannitol salt agar is that it is both selective, as we've been talking about, and also differential. Remember, those were the two types of special media that we introduced. Selective media, which is going to select for and against certain organisms. And then differential, which is really going to help us differentiate, hence the name, certain strains. So it turns out that mannitol salt agar is both selective and differential. We'll talk about that in just a couple minutes. Differential media, like the name suggests, differentiates between strains of bacteria by indicating whether certain metabolic products are being made. And it's the differential media that typically have that indicator dye in it. So mannitol salt agar is differential. I've listed a couple other very common differential media here. There's one called Mcconkey Agar, and that one I mentioned earlier, blood agar. Now, Mcconkey Agar is going to select specifically, in other words, it's going to allow to grow gram-negative enteric bacteria. So gram-positives can't grow on Mcconkey because of what's in it. That's selective, and it's going to differentiate certain kinds of enteric bacteria. That word enteric just means intestinal. Now, the way that Mcconkey is differential is right here. It's going to tell us the difference between lactose fermenters and non-lactose fermenters. In other words, which of these gram-negative enteric bacteria can ferment lactose and which ones can't? So Mcconkey is selective and differential. It selects for gram-negative enteric bacteria. All the other bacteria cannot grow on it. And it differentiates the lactose fermenters. It's going to tell us if we have a lactose fermenter. What happens is, on Mcconkey Agar, if the bacterium that is growing, this gram-negative intestinal bacteria, if it's fermenting the lactose that's inside the media, it's going to start creating acidic byproducts. That's going to cause the colonies to turn pink. There's a dye, an indicator dye in the media, and it's going to cause the colonies to turn a pink color. If we have a gram-negative intestinal bacterium that can't ferment lactose, that means those bacteria are not going to change the pH to acidic, and we're going to be left with a neutral pH. And those colonies are going to grow just as ordinary, pale, beigeish, colorless colonies. So Mcconkey's has both characteristics. It's going to be selective and differential for different strains of gram-negative enteric bacteria. Blood agar is a differential agar. Now, here's something to know about blood agar. Blood agar is not selective. Just about anything can grow on blood agar because it's complex. Remember, it's one of these enriched media. It's got blood in it, and blood is a very nutritional substance. So just about anything can grow on blood agar. There's no selection going on, but there is a differentiation. Streptococcus species have a characteristic sometimes of being able to hemolyze or break down red blood cells. You can imagine that if you've got a bacteria in your body that's capable of breaking down your red blood cells, you're in trouble. That's a pathogen. So what the blood agar allows us to do is see whether or not we've got a strain of Streptococcus that is capable of breaking apart or hemolyzing red blood cells. Those strains have an enzyme called hemolysin. Hemolysin is what breaks apart the red blood cell. So if we were going to categorize blood agar, we would call it complex or enriched, and we would also call it differential. It's going to differentiate strains of Streptococcus that can break apart red blood cells. Now, We've talked about chemically undefined or basal media. We've talked about complex or enriched media. We've talked about selective media, differential media. There are two more categories: transport and storage. We've already talked about transport media, right? Transport media is used to literally transport cells from one place to another, usually from where the cells are being collected, like at bedside in a hospital, to where those cells are going to be analyzed, the microbiology laboratory. Remember, transport media does not encourage the growth of cells. It has just enough nutrition in it to keep the cells alive during the transport period. It just has enough nutrition to keep those cells alive, but it's not going to encourage them to grow. Now, can anybody imagine why we wouldn't want the cells in a patient sample to grow on the way to the lab? Any ideas? Why would I not want, let's say, to swab that abscess on my patient, put it into the tube, and let's say I'm going to send it for, I don't know, a 6-hour ride to the Quest lab or the LabCorp lab? Why would I not want to let those cells start dividing and growing? Any ideas? So Carrie's saying you don't want to get a sample that appears more virulent and widespread than it actually is. Now that term virulent means something that very specific and not what I think you're thinking of right now. What you don't want, like Carrie's saying, is you don't want it to arrive at the lab appearing to be more, appearing to have more cells than it really did, right? If I swab an abscess and I send it to the lab and it gets there and it is just teeming with bacteria, that's a different thing than if it gets to the lab and there are a few bacterial cells in there. Prognostically, right? It's a different thing if you've got an abscess that is just loaded with bacteria versus one that has, you know, a lesser number of bacteria in it. It's the difference between an abscess that is sort of under control by the immune system and one that's not. So you don't want the sample to appear as though it had more cells in it than it really did. The other thing that might not be as obvious is sometimes in something like an abscess, there's going to be one genus and species of bacteria that's causing the problem. But when you sample it, you're also going to get a couple of other species in there that aren't causing the problem. They just happen to live on the skin. Now, the way we know one from the other is by the number. A good example of this is with urine samples. Most urine samples are collected clean catch, right? We send the patient into the bathroom with a little sterile cup and a couple of alcohol pads, and we tell them to clean themselves off and then pee into the cup. It's very common to find bacteria and fungi, yeast, in a clean catch urine sample, but at very low numbers. We know if we see a few yeast, a couple of E. coli, that person does not have a bladder infection. So cell number matters. In comparison, if that person pees into a cup, clean catch, and the sample is just teeming with E. coli, I know I have a patient with a bladder infection. So cell number really matters. We don't want those cells to start dividing on the way to the lab, because that's going to give us an abnormal sense of how many cells were really in the patient. Like Valerie is saying, you want a true sample. You want an accurate reflection of what's in the patient. You don't want to allow it to artificially divide over time. Like Elaine's saying, an accurate baseline. If we allowed it to divide, it would give us a false sense of what was happening in the patient. Good, very good. So that's our transport media. The last category, number 6, is storage media. Now, storage media is probably more commonly used in research laboratories than in clinical laboratories, but we should know about it either way. Storage media, like the name suggests, is a medium that we're going to use if we want to save a sample of cells for the long term. If you're not familiar, we cannot put a living cell into a freezer and then thaw it and have it be alive. You just can't do it. When you freeze a cell, what happens is the water inside the cell is going to expand. Remember, water has that very unusual characteristic. It expands when it becomes a solid, and it contracts as a liquid. So if we freeze a cell, the water will expand and the membrane will break. We're going to destroy it, essentially. When we thaw that cell and the water melts, well, all that's going to be left is pieces of the cell. So if you want to freeze a cell and be able to thaw it later and have it be alive, you have to get the water out of the cell and replace it with another liquid. And the molecule that we use is called glycerol. At least the molecule we use most often is called glycerol. If you put living cells into a glycerol solution, the glycerol will come into the cell across the membrane and it will push the water out. Then you can freeze that cell because the glycerol will not expand as it freezes. Then when you thaw the cells, all you have to do is let the glycerol thaw out, and then you put the cells back into a nice saline solution, and the saline and the glycerol will switch places. So storage media is going to have that glycerol in it so that we can freeze microbial cells and keep them for the long term. And by long term, sometimes it's weeks, months, but you can keep cells for years and years. I think I mentioned to you in my bio that my PhD work was in human physiology and specifically reproductive. I'm very interested in reproductive medicine. And one of the things that we did in our lab was we worked with embryos, cattle embryos, not human, that were very young. They were only a few days old. So what we call pre-implantation. They had not yet implanted in the uterus. And we often would freeze those embryos. But we had to use a solution that had glycerol in it in order to have the embryo survive. Nowadays, in reproductive medicine for humans, it's very common to freeze embryos. We put them on dry ice. We put them in liquid nitrogen, and we can save them for years and years and years. There's some ethical questions around that. There have been legal cases where people have frozen embryos over time and then not known what to do with them because they didn't want another child and they had these embryos frozen. And at some point you have to decide, should I keep them? Should I donate them? Should I destroy them? So there's lots of ethical questions over long-term storage of embryos, but we're capable of it. We're capable of storing living cells in a freezer for long, long periods of time and have those cells still be alive after we thaw them out. Okay, So those are our 6 categories of media. Does anybody have any questions? Does anybody have any questions about the types of media, the difference between types of media? Remember, we have all of these at our disposal. And within each of those 6 categories, you know, there are lots of different recipes. So there's not one chemically defined media. There are lots of them. There are lots of complex media. There are many different selective media, many different differential media. So we have lots to choose from, which means we have to understand what it is we're trying to accomplish. It would be really hard to try to grow microbes without knowing a little bit about the situation. If I know that a sample came from a recreational body of water, I have some idea of what might be in it. If I know the sample came from a hospital room, I have some idea what might be in it. And I can use that knowledge, that history, if you will, to pick and choose what media I'm going to use in order to try to identify that organism or to do something with it for my own needs. All right. So we're going to switch gears now and we're going to do a little exercise. Now, this exercise is fairly complex compared to the other things that we've done so far. We're going to be examining 4 different types of bacteria. You'll notice that I call them bugs. That's just something I picked up in veterinary practice. I often refer to bacteria as bugs. The first organism we're going to be using in our experiment is our old friend E. coli. Remember that E in E. coli, it stands for something. The genus is Escherichia coli. Almost everybody calls it E. coli. Escherichia is a miserable word to spell. E. coli is a gram-negative rod. That's its gram status. We also know that E. coli is one of these enteric organisms, or what are sometimes also called fecal organisms. It lives in the intestine of certain animals, primarily mammals. And we refer to it as being fecal because it's going to come out of the animal in the feces. And the way that pathogenic E. coli tends to spread is through feces. So E. coli is our first organism of the day. Our second organism is called Enterococcus Fecalis. This one is a gram-positive coccus, so it has a very different gram status, but it's also enteric/fecal. Some people, especially in older source material, referred to this as a fecal streptococcus. From time to time, I'm going to give you old terminology that we're not using anymore. And the only reason I give it to you is because you will find source materials that are older. You'll find old textbooks in hospitals, for example. So if you ever hear something referred to as fecal streptococci, we now call that Enterococcus. Entero, again, meaning intestinal. It's an Enterococcus. Third on the list is Staphylococcus Epidermidis, that organism we just talked about. That one is a gram-positive coccus, and it tends to be located on the skin. Number 4 is Staphylococcus aureus, the other one we talked about. Again, gram-positive coccus, likes to live on the skin, in the nose, and in the pharynx, the back of the throat, in other words. So all 4 of these organisms are normally found in the human body, all 4 of them. But there are some strains that are pathogenic. Now, we're going to talk about these 4 organisms and how they grow on special media, but the experimental part of this is at number 5. What I'm going to do, or what I've done, is I chose 2 of these and I mixed them together in one culture container. So our goal today is to figure out if we can tell which 2 I put into our unknown mix by looking at how they grow on special media. Now, today's exercise is also going to use a couple of different kinds of culture media. We're going to grow all 4 organisms on nutrient agar. Remember, that's one of those basal media. We're also going to grow them on mannitol salt agar, 7.5%. We're going to try to grow them on that agar. And if I go ahead just a bit, we're going to try to grow them on eosin methylene blue agar, which we haven't talked about yet. That one's called EMB. Now, mannitol salt agar, as we talked about just a minute ago, is a selective and a differential type of media. It selects for 2 kinds of Staphylococcus, at least if we put 7.5% salt in it. It's going to select for Staphylococcus Aureus and Staphylococcus Epidermidis. It's going to select against virtually everything else. Although remember, it is possible to get a weak growth of Enterococcus and Micrococcus. And of course, one of our organisms today is an Enterococcus. So of our 4 microbes, E. coli, Enterococcus, Staph Epidermidis, Staph Aureus, 2 of them should grow well on MSA. 1 of them should grow poorly on MSA. And 1 of them shouldn't be able to grow on it at all. Now, we haven't yet talked about how MSA is differential. The reason that it's a differential media is because it contains something called mannitol. It's in the name. It contains mannitol and it contains sodium chloride as the salt. We know what the salt does. The salt helps it select. But the mannitol is differential. Mannitol is a carbohydrate. It's a sugar. And some bacteria are able to ferment mannitol. So this media will differentiate for us organisms that can ferment mannitol from organisms that cannot ferment mannitol. And guess what? The 2 things that grow on MSA, 7.5%, 1 of them is a fermenter and 1 of them isn't. Staphylococcus aureus is a mannitol fermenter. So as it grows on the MSA, it's going to consume the mannitol that's in the media, and it's going to ferment it. It's going to produce an acid, and that acid is going to lower the pH of the media. There's an indicator dye in MSA, and when that indicator dye is in an acidic pH, it turns yellow. MSA starts off as this sort of a pinkish red color. This is what an MSA plate looks like when it starts off. So it's ordinarily this pinkish color. It's not a deep red like blood agar is. It's more of a pinkish red. Staph aureus can grow on this stuff because it doesn't mind that high salt concentration. When it grows, it's going to ferment the mannitol that's in this media. It's going to make an acidic byproduct. It's going to lower the pH of the media, and the media is going to turn yellow. Staph epidermidis can grow on MSA, but it doesn't ferment mannitol. So when Staph epidermidis grows, the media is going to stay pink. So we literally have a visual way to tell which one of those two species is growing. Only two species can grow well on this media. One of them can ferment mannitol and one of them can't. So all we have to do is look at the plate the day after we've incubated it, and we're going to know, do I have Staph Aureus? Do I have Staph epidermidis? On this slide, over on the left, you're looking at an MSA plate before it's inoculated. Again, sort of a pinkish red color to the auger. Over here on the right, we're looking at an MSA plate that was inoculated with two organisms. We took some Staph epidermidis and we streaked, you can see the streak lines right where I'm pointing to. We streaked some Staph epidermidis cells onto this side of the plate. So we knew we had Staph epidermidis. And down here, we streaked some Staph Aureus cells on this half of the plate. We incubated this plate for 24 hours. And look what comes out of the incubator. There's growth on both sides of the plate, isn't there? I can see this sort of dark or darker yellow color where the cells are growing. I don't see colonies, I see these streak lines. There's lots and lots and lots of cells growing on these lines. So no individual colonies here, but I wasn't trying to grow individual colonies. I just wanted to grow cells, period. Same thing up here. I can see growth on these darker pink lines. So both of these organisms grew, but only one of them fermented the mannitol, the Staph Aureus. I know because it turned yellow. Staph epidermidis, it'll grow, but it can't ferment that mannitol. It cannot ferment that carbohydrate called mannitol. So the media stays that original pink color. All right. Now we're going to talk about this medium called EMB, eosin methylene blue. We've actually mentioned methylene blue before. Methylene blue is one of the stains we use. We looked at some cells that were stained with methylene blue early on in the course. Remember, I told you that methylene blue is, like all stains, is toxic to cells, but EMB has a very particular, very low quantity of methylene blue in it. So there are some organisms that will grow and some that won't. It is selective. EMB is selective for E. coli and other fecal coliforms. Now, we haven't mentioned this term coliform yet. A coliform is any bacterium that grows in the intestine that can also ferment lactose. So if it grows in the intestine, so it's enteric or fecal, and it can ferment lactose, we're going to call it a coliform. That's an important term to know because on a lot of environmental sampling reports, like water quality reports, what you're going to get back in the results is whether or not there are coliforms present. They're not going to tell you specifically that it's E. coli, which is one of the coliforms, but just that there are coliforms. Remember, we talked about the event in Burlington, Massachusetts that happened last week. When they sampled the water in the storage tanks, they discovered coliforms in it. Now, was it definitely a pathogenic E. coli? Nobody knows. We didn't do that. They didn't do that kind of testing. But when we see coliforms, we get concerned because some coliforms are pathogens. Not all of them. Most of them are not pathogenic, but some of them are. So out of an abundance of caution, when we see coliforms in water, we don't drink it. We don't bathe in it. We don't brush our teeth with it because we don't want to ingest a pathogenic coliform. So Stephanie's asking, so Staph epidermidis cannot be present if the color is yellow. That is correct, Stephanie. If it grows on MSA, I'm going to change that. If it grows well on mannitol salt auger, it is either Staph aureus or Staph epidermidis. We tell those apart by the color. If it grows well, it's one of those two. If it turns the media yellow, it's Staph aureus. If it leaves the media pink, it's Staph epidermidis. Diagnostic. Okay. So, E. coli and other fecal coliforms, these are all gram-negative organisms. Most of them are gram-negative rods. Not all of them, but most of them. So EMB is going to select against anything that's gram-positive. And that includes that organism we're using today called Enterococcus Fecalis. Remember, if I go back to this slide, Enterococcus Fecalis does live in the intestine, but it's a gram-positive organism. E. coli is gram-negative. So there are organisms living in our intestine that are gram-positives, and they will not grow on EMB. Only gram-negatives will grow on EMB, and only these E. coli and fecal coliforms, as well as some other non-coliforms. So I'm going to add some things in here in real time, just so if you go back to this, it will make sense. These are gram-negatives, gram-negative organisms, specifically E. coli and other fecal coliforms, as well as some other non-coliforms, as long as they are gram-negatives. Now, EMB, just like MSA, it's both selective and differential. It's differential because it can tell the difference between a lactose fermenter and a lactose non-fermenter. There's a second sugar in EMB called sucrose. So again, if an organism can ferment sucrose versus if an organism can't ferment sucrose. Now here's where it gets a little confusing. EMB is going to show us if it ferments one or the other or both. As long as it ferments one of them, you're going to see a change in the media. So it's lactose and or sucrose. Some E. coli, some fecal coliforms, they can ferment both sugars. Some of them can only ferment lactose. Some of them can only ferment sucrose. We put both sugars in because it's going to allow us to cast the widest net when we're trying to identify the organism. So How does it work? Well, if the organism is fermenting one of these sugars or both of them, we're going to get purple colonies with a very distinct green metallic sheen. And this really is a metallic green color. If you see a purple colony growing on EMB and it has a metallic green color when you hold it to the light, it's E. coli. Let me say that again. If it grows in colonies or streaks and it's purple, the colonies have to be purple. And when you sort of tilt the plate in the light, you get this green metallic color. That's E. coli. That's nice, right? If I have a sample and I want to know, is there any E. coli growing in this? I can streak it on EMB, I can incubate it overnight, and I can know. Now, I'm not going to know what strain of E. coli it is. I'm not going to know if it's a very dangerous pathogenic E. coli or if it's just plain old E. coli that lives in our gut anyway. But I'll know it's E. coli. Now, the second thing I might see is a purple colony with no green color. If I get a purple colony, but there's no green metallic sheen, that's telling me I've definitely got a gram-negative. I've definitely got an intestinal or a fecal organism because it's growing. But if it doesn't have a sheen, I know it's not E. coli. And finally, if I see a colony growing on EMB that's that plain old sort of white or beige color, or even colorless, that tells me the organism is not fermenting the sugars. So the purple color that I see in organisms that grow, that purple color comes from the fermenting of the sugars. If it grows purple, I know it's fermenting. If it grows purple and green, I know it's E. coli. If it grows as a white, beige, sort of colorless colony, I know I've got a gram-negative intestinal organism that doesn't ferment those sugars. And there are a couple of big players that fall into that category: Salmonella and Shigella. Those are two genuses of bacteria. They will grow on EMB because they're gram-negative, but they don't ferment those sugars, so they are not coliforms. Let's take a look. On this slide, in the upper left-hand side, this is what an EMB plate looks like before it's inoculated. It has a really distinct color. I like to refer to it as a grape juice color. That's just what my eyes see. It does not have grape juice in it, but it has this sort of it's a purple color, but it falls into the purple grape category for my eyes. Now look at this plate over here on the right. This, by the way, is a quadrant streak plate. Somebody took a sample of cells and spread them out over 4 quadrants. So they took the cells out of, let's say, a broth tube, and in the first quadrant, they just smeared them. That's where you're going to get your heaviest growth. Then they cleaned off their loop. They sterilized their loop. You draw your loop through that heavy growth area, and then you streak again. So in other words, I'm going to grab some of these cells with my loop and I'm going to spread them out. Then I sterilize my loop again. I draw it through those and I streak and spread them out. And then I do it a fourth time. Sterilize my loop, draw through that quadrant and spread them out. Quadrant streak plating is designed to create individual colonies. Sometimes you won't see any colonies until you get to the fourth quadrant. Sometimes you'll get them in the third quadrant, like you can see here. Sometimes you'll get them in the second quadrant. It all depends on how many cells were in the original sample. But the idea is by pulling the cells from one quadrant into another and then streaking them out, I'm pulling fewer and fewer and fewer cells because I want to get to the point where I am separating the cells on the surface of the agar enough that I'll see individual colonies growing. So this is a plate an EMB plate that is classic for E. coli. You can't even see the purple because there's so much green on top of it. See that green metallic color? It's really distinct, isn't it? You wouldn't mistake that. That's not a hard thing to see on an EMB plate. If there is a green metallic color, you're going to see it. Let's take a look when we don't see that green metallic color. Look at the plate here on the lower left-hand side. This is a plate that was divided into 4 quadrants. They used a Sharpie pen. We used a Sharpie pen on the bottom of the plate so that we could see 4 quadrants. And we put 4 different organisms on this plate. So we dipped the inoculating loop into culture and just smeared the cells one time. So I have just one big smear of lots and lots and lots of cells right here. I don't have individual colonies. I just have this area of growth. So this is E. coli. It's growing, so I know it's gram-negative, and I know it's enteric. I know it's an intestinal organism. I also know that it's fermenting lactose and or sucrose. So I know it's a coliform because coliforms are fermenters. And the reason I know it's E. coli is because it has that green color. Over here in this streak, this is that purple color I was talking about. These cells are growing purple on this agar, but they don't have that green metallic sheen. That tells me they are gram-negative because they're growing, and they are intestinal organisms, and they are coliforms. They are fermenting those sugars because that's what makes them purple. The E. coli really is purple underneath that green metallic color. There's purple. You just can't see it very well. So coliform, specifically E. coli. Coliform, something other than E. coli. Down here, see this streak? This is what Salmonella looks like, or Shigella. This is a gram-negative intestinal organism that is not a coliform. No purple color. It's growing, so I know it's gram-negative, but no purple color. So there's no fermentation going on of the sugars. Salmonella and Shigella are 2 common pathogens in our intestine that are not coliforms. And finally, over here in the 4th quadrant, I streaked a gram-positive organism. No growth. Gram-positives cannot grow on EMB. Can't do it. So it's selective for gram-negative organisms that grow in the intestine, and it's differential for E. coli and other coliforms. It's going to differentiate them from non-coliforms based on the color. I can also use EMB to identify E. coli because only E. coli is going to give me that green sheen on top. It's a very useful little media, isn't it? It's very helpful. It's going to tell me a few things right away. It's going to help me break down what I might be dealing with right away. Okay, now I'm going to show you one other thing that you might see on EMB. Not to confuse you, not to upset you. It's just a little thing that sometimes we see on EMB. And it's what we refer to as fish eye colonies. Take a look at the plate on the left first. This is a quadrant streak plate, isn't it? I can see that the person first just streaked a whole bunch of cells, then pulled a few of those cells into the second quadrant and streaked those, then pulled a few of those cells into the third quadrant, streaked those, then pulled some of those into the fourth quadrant and streaked those. And in that fourth quadrant, I got colonies. That's what I was trying to do. I was trying to get colonies. You can really see on this plate that the colonies are purple. Now, this happens to be E. coli. Notice where the green color is most prominent. It's most prominent in the areas of heavy growth, millions of cells. You don't see the green sheen on these individual colonies, do you? You can see the purple color much better, but you got to look in the areas of very heavy growth to see the green metallic sheen. This is E. coli. And I will get a green color for E. coli, but only in areas where the growth is heavy, where I've got lots and lots and lots of cells growing. So that's one thing to note. The other thing to note is this. Sometimes these purple colonies have a very distinct black center. Can you see that? Sometimes when E. coli grows on EMB, it'll develop these really weird black centers. These are called fish eyes. Somebody looked at this and thought about a fish and a fish's eye. Again, it doesn't happen with all strains of E. coli, but you might see it. You might see fish eyes, and that's an indication that you have a coliform, not specifically E. coli, but you've got a coliform if you see fish eyes. And all a coliform is an intestinal organism that is able to ferment lactose and or sucrose. Okay, so remember what we're doing today. We've got 4 organisms. We've got E. coli, Enterococcus Fecalis, Staph Aureus, Staph Epidermidis. I took 2 of those secretly. I took 2 of them and I mixed them together in one container. And I cultured those cells. And then I used those to inoculate nutrient agar, mannitol salt agar, and EMB. Question number 1. Why did I bother using nutrient agar? Nutrient agar is a basal media. It's going to grow virtually all non-fastidious organisms. Why would I take the time when I'm doing an experiment to take my 4 different organisms and smear them onto some nutrient agar? Why would I do that? I'm really interested in whether or not they grow on the special media, right? That's what's going to help me figure out what the secret mix is. So why would I bother taking those 4 organisms, streaking them out on a nutrient agar plate and putting that plate in the incubator? What information is that going to tell me? Carrie's got it. Elaine's got it. When we're working in the laboratory, when we're doing experimental work in the laboratory, and we're using these cultures that we grow, if I'm going to proceed with an experiment, the very first thing I need to know is, do I have growing, healthy, living cultures? I have these 4 tubes that have been sitting in the incubator. I think I have E. coli, Enterococcus, Staph Aureus, Staph Epidermidis in my 4 tubes, but I need to make sure they're all alive before I start using them. If they're alive, they're going to grow on nutrient agar. It's a control. It's a control. Remember, in the microbiology lab, we're working with living things. So if I want to experiment with them, I have to first show that they're alive. What I wouldn't want to happen is I start playing around with special media and I say, oh, look, it didn't grow. It didn't grow, so therefore it must be this. Well, either it's that or it wasn't alive to begin with. I have to check. I have to double check that all of my cultures are alive before I start playing with them in the laboratory, before I start using special media. I have to show that they are alive and capable of growing before I start putting them on media that may or may not allow them to grow. Imagine if I had a selective media. Imagine if I had an organism that I knew couldn't grow on MSA. And I took some of that organism and I put it on MSA and I put it into the incubator, and the next day there was no growth. Is it because the salt killed the organism, or is it because the organism was dead to start with? I don't know. I don't know unless I include a nutrient agar plate. If I can show that it grew on a nutrient agar plate, but it didn't grow on the MSA, now I have some data. Make sense? It's a control. When we work with living organisms, we have to first show that they are alive and capable of growth. And then we can start playing with them in the laboratory. Okay. So what I did was I took my nutrient agar plate and I drew 4 quadrants basically with a Sharpie on the bottom of the plate. And then I wanted to transfer cells out of 4 culture tubes, 4 broth tubes that had my cells growing in it. I had 1 tube for E. coli, 1 for Enterococcus, 1 for Staph Aureus, and 1 for Staph Epidermidis. So using my very best aseptic technique, I made a very simple streak pattern onto each of the onto each quadrant of that nutrient agar plate. I don't need to grow colonies here. I just need to show that the cells are alive and they can grow. I don't need to grow isolated colonies. I'm not going to do colony morphology. I don't have to count colonies. I just want to make sure that they're alive and capable of growing. I took that plate with 1 organism in each quadrant. I put it upside down in the incubator, of course, at 35 degrees centigrade for 24 hours. Remember, 35 to 37 degrees is the most common temperature we use in the microbiology lab. Came back 24 hours and I examined the results. Now remember, the other thing I did was I secretly chose 2 of those 4 organisms and I combined them into what I'm going to call the unknown mixed culture. So here are nutrient agar plates. Now these are 2 plates that students made. But you can see what I mean by we drew with Sharpie on the bottom of the agar plate to create these 4 quadrants. And then what the student did was just smear cells. 1 for E. coli, I'm just guessing. 1 for E. coli, 1 for Enterococcus, Staph Aureus, Staph Epidermidis. All 4 of them grew very well on the nutrient agar, just like we would expect them to. Remember, nutrient agar is a basal media. It's going to allow most non-fastidious bacteria to grow. But I'm not going to be able to tell anything from them. I'm not going to see any special colors or anything. I'm just going to be able to see that they grew. This student did the same thing. This student wasn't quite as careful with their streak lines. You can see they sort of bumped into each other on the streak lines, but that's okay because all this plate needed to do was show growth. As long as I could see growth, I knew I had 4 living bacterial cultures. You can see where this student drew the quadrant lines and labeled each quadrant. Notice that this student didn't label, very naughty. Remember, anytime you're working with microorganisms, you've got to label, 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 so that you know exactly where things are on your plates. Okay. So I chose 2 of the 4 organisms and I made an unknown mixed culture. For the second part of this experiment, what I did was I took 1 MSA plate and I took 1 EMB plate. Only 1 of each because I took those 2 organisms and I mixed them together in 1 culture. So I did a quadrant streak on MSA and I did a quadrant streak on EMB. Both organisms mixed together. This diagram over here just shows me again the pattern for a quadrant streak. I put the loop full of cells out of the culture tube into quadrant 1 and I just smear it around. Then I flame my loop or incinerate my loop to sterilize it. I'm going to pull some cells out of there and I'm going to spread them around quadrant 2, flame my loop, pull some cells out of here and spread them, pull some cells out of there and spread them, and hopefully I'll get isolated colonies. Here are our results. This is the MSA plate. It's very difficult to see, I know, but there are individual colonies growing on this plate. It's not perfect, but I can see growth in this quadrant here and in this quadrant here. It turns out this was quadrant 1 and this was quadrant 2. Remember, sometimes you won't see colonies until you get all the way to quadrant 4, but it depends on how many cells were in the original culture. In this case, there were few enough cells that I could see individual colonies even in the second quadrant. Now, this is what I want you to see on this plate. First of all, there's only 1 type of colony on this plate. And second of all, that colony growth turned the media yellow. There's 1 thing growing on this plate, and it turned the media yellow. I have some important information, don't I? There are 2 bacteria in my unknown mixed culture. Only 1 of them grew on this plate. The one that grew turned the media yellow. What is the organism that grew on that plate? What do we know can grow on MSA and turn the media yellow? Type it right into chat for me. There are only a couple things that can grow and grow well on MSA. Those were growing well. Those were good-sized colonies. There's only a couple things that'll grow on MSA. And there's only one thing that will turn it yellow. Good. Connie's got it. Stephanie, Mackenzie, Marilyn, Valerie, Carrie, everybody's got it. Good. Staphylococcus aureus. One of the two organisms that I chose and put into my mixed unknown culture is Staphylococcus aureus, for sure. But I don't know what the second one was. I don't know yet. I do know some things though, don't I? I know some other things, don't I? Could the second organism be Staphylococcus Epidermidis? Could the unknown mixed culture be Staph Aureus and Staph Epidermidis? What do you think? I did a quadrant streak. I got lots of individual colonies. But all of the colonies turn the media yellow. What do you think? Is there Staph Epidermidis in that mixed unknown culture? Why or why not? Elaine's saying no, it can't. There can't be any Staph Epidermidis in there. What do you think? Let me ask you this: if the second organism is Staph Epidermidis, how does Staph Epidermidis grow on MSA? How does it grow? What do I expect it to look like when Staph Epidermidis grows on Mannitol salt agar? What would I expect to see? Connie's got it. Elaine's got it. Remember that plate I showed you, the MSA plate, where we divided it in half and we streaked Staph Aureus on one side and we streaked Staph Epidermidis on the other side? Staph Epidermidis grows on MSA, but it leaves the media that pink color. Just a small note, Elaine. It doesn't turn the media pink. It leaves the media pink. It doesn't change the color of the media. So if there was Staph Epidermidis in my mixed unknown culture, I should have seen some colonies growing on top of pink media. But all of those colonies that were growing turned the media yellow. So I know I don't have Staph Epidermidis as my second organism. So I know Staph Aureus is in there. I know Staph Epidermidis is not in there. I still got to find out what the second one is. It's either E. coli or it's Enterococcus Fecalis. We need an EMB plate. We need an EMB plate to figure this out. Here is my EMB plate. And yes, this plate was inoculated. This plate was inoculated with a quadrant streak of the mixed culture. I turned it upside down. I put it in the incubator. I left it overnight. And this is what I got. There's nothing growing on this plate. Not a thing. Not a thing growing on this plate. Sometimes when you use special media, you have to interpret negative results. You have to figure out what it means when something doesn't grow. I know that one of my organisms is Staph Aureus. I don't know the other one though. I know it's either E. coli or it's Enterococcus Fecalis. I got no growth on EMB. How do I interpret that? Elaine's got it. Mackenzie's got it. Carrie's got it. Connie's got it. Is it E. coli? It's not E. coli, is it? E. coli grows great on EMB. It grows as a purple colony because it ferments lactose and or sucrose. And it has a green metallic sheen when it grows. I didn't get any growth. Enterococcus Fecalis lives in our intestine, but it's a gram-positive organism. And EMB selects against gram-positive organisms. I'm left with Enterococcus Fecalis. Enterococcus Fecalis shouldn't grow on EMB, and it didn't. Staphylococcus Aureus also shouldn't grow on EMB, and it didn't. Both of those are gram-positive organisms. And EMB selects against those. It won't allow them to grow. So sometimes we have to interpret a negative result. We have to interpret what it means that something didn't grow. Because with any selective or differential media, there are some things that will grow and some things that won't. And that's exactly right, Carrie. That's why we use the control in the beginning. If I pulled that EMB plate out of the incubator and I look down at that and there's nothing growing, how can I say, oh, well, it must be the gram-positives that grow on here? How can I say that unless I've shown that my gram-positives are alive and healthy and capable of growing? I need that nutrient agar plate, because that's going to show me that they're perfectly capable of growing, but they can't grow on EMB because EMB selects against them. EMB will not allow gram-positives to grow at all. So I can go down my list and I can tell exactly what I have based on those results. It can't be E. coli. It simply cannot be E. coli, because if it was E. coli, it would have grown on EMB and it would have had a green sheen to it. Purple colony, green sheen. If I only had the MSA plate though, the MSA plate, I wouldn't know. Maybe it was E. coli, maybe it wasn't, because E. coli shouldn't grow on MSA. I have to have the combination of the MSA plate and the EMB plate to be able to say it's not E. coli. E. coli should not grow on MSA, but it should grow on EMB. And whatever I had didn't grow. So I crossed E. coli off the list. I could also cross Staph Epidermidis off the list because While I wouldn't expect Staph epidermidis to grow on EMB, I would expect it to grow on MSA. And I would expect it to leave the media pink. And what grew on my MSA plate turned the media yellow. So I knew I didn't have Staph epidermidis. Make sense? Special media. Special media helps us select for, select against, sort of pick and choose what we can grow, and differentiate among those, closely related species, those different strains. It allows us to do some detective work and narrow down what we think we have. Any questions? All right, everybody. So you are ready now to do your homework for the week. Fungi and special media. Keep up with your lectures. Keep up with your lecture quizzes. And I will see you on Monday. And get outside and enjoy this weather. It's not too hot. All right, everybody. We'll see you next time.
Thank you.